And speaking of science, one of your most noted accomplishments is your development of the paper-based diagnostics for Zika and Ebola. So regarding this, intriguing discovery, can you explain the whole process and its significance?
Yeah, it grew out of our work in synthetic biology, going back to the late 90s, so probably around the time you and many of your listeners were born, Karen. We recognized that you could introduce engineering principles at the molecular level, and we, along with several other labs, began to advance what became synthetic biology, this idea you can use engineering principles to model, design, and build synthetic gene circuits and other molecular components, and then use these circuits and components to rewire living cells, endowing them with novel functions for a variety of applications. Yeah, so going back now 7 years ago, following a lot of efforts on engineering synthetic gene circuits and using these inside living cells, Keith Pardee, a postdoc in my lab, began to play with cell-free systems. What are these? This is this idea that you can open up a living cell, remove the machinery of that living cell, which consists of DNA, RNA, ribosomes, other molecular machines, molecular components of molecules like ATP, nucleotides, and play with them in petri dishes and test tubes. This was not new. This has been done for decades before I was born in molecular biology. And Marshall Nirenberg, for example, worked out the RNA code before I was born using cell-free systems. Keith's advance first was to show that you could introduce synthetic gene circuits into cell-free systems and they could function. You could have transcription and translation. But his key discovery was that he demonstrated that you could take a cell-free system along with a synthetic biology construct, spot it onto paper, freeze dry it, and then sometime later rehydrate that cell-free extract along with the synthetic biology component circuit and reactivate it so that it would function as if it was inside a living cell, but now it's been embedded into paper. And so it could perform transcription and translation. And we got very excited about a number of applications, the most dominant being that we could use this to create low-cost, easy-to-transport paper-based diagnostics, wherein we could embed cell-free extracts along with various synthetic biology sensors onto paper that could then be taken out into the field in low-resource settings without need for refrigeration. So you can just stick it in your pocket, get on a plane, take it around the world, use it, stick it in a regular envelope, send it around the world, use it, and use it to test for bacterial pathogens, viral pathogens, parasites, as well as human host cell responses.
Yeah, and so regarding this paper-based diagnostics, there's also COVID-19 going on. So would the paper-based diagnostics work for COVID-19 or any other type of disease?
They do. So Keith Pardee has launched a company called Encarta using some of our earlier sensors to address COVID-19. And going back a few years ago, we also demonstrated, in addition to synthetic biology elements, you could embed CRISPR elements into paper and other cell-free settings. And we launched a company called Sherlock Biosciences that pivoted in the beginning of the pandemic to actually produce the first CRISPR-based FDA-approved product, which was a COVID diagnostic. It's used in clinical labs, CLIA-based labs. They've now partnered with 5 different global diagnostic firms that are on pace to do 10 million tests per month using this test.
Drunk mistakes, we're bound to hit the